out of the two structures, किसका development starts first? Endosperm का development starts first. The primary endosperm cell, PEC, right? Now, endosperm development, as I said, happens in three ways. Nuclear, cellular, and halobial method of development. Nuclear and cellular have been mentioned in the NCERT. Halobial is not mentioned, so I will delete it from here. Better not to write it. Barabar? What is the starting point always? PEC. Starting point always will be PEC. So, PEC will start undergoing mitotic division, but only karyokinesis. कौन सा टाइप का डिवीजन है ये? फ्री न्यूक्लियर डिवीजन. बराबर ना? सो karyokinesis, 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 karyo, रिजल्टिंग इन मल्टीपल न्यूक्लियर. तो इसलिए ऐसे एंडोस्पर्म को हम लोग क्या बोलते हैं? न्यूक्लियर एंडोस्पर्म. सारे karyokinesis होने के बाद फाइनली इट विल स्टार्ट फॉर्म साइटोकाइनेसिस. इट विल फॉर्म साइटोकाइनेसिस. तो सेल वॉल्स विल बी लेड डाउन देयर. सेल्यूलर में PEC will undergo karyo followed by cyto. Another karyo followed by cyto. Is the difference understood? तो न्यूक्लियर में कैसा होगा? All karyo first, then followed by all cyto. सेलुलर में क्या होगा? Karyokinesis, cyto. Karyo, cyto. So, will this give rise to a structure wherein you will have cell wall formation now? बराबर ना? तो अल्टीमेटली ये वाला भी तो बनेगा, तो यही ना? यानी न्यूक्लियर भी अल्टीमेटली साइटोकाइनेसिस होने के बाद तो सेलुलर ही बनेगा, बट द पाथ टेकन इज डिफरेंट. The path taken is different. So both of them ultimately reach the same goal. Right? Now to give this kind of, see, nuclear is the most commonest way of development of endosperm. Now to explain both of them, they give a very good example. Coconut. What type of coconut? Fruit क्या? Coconut का fruit कौन सा है? Drupe. Right? Now coconut is a drupe. Now they have given in a tender coconut. Tender coconut is something which is water. पानी के साथ वाला. Right? Now the liquid part is nothing but nuclear endosperm. And the malachite, yes, white kernel it is called as malachite. Right? Now, that is cellular endosperm. Right? So if we take a tender coconut which is full of water, the whitish part is very thin, right? But then as time passes, gradually the white part starts getting thicker and thicker. So what is happening? The nuclear endosperm is gradually converting to cellular part. समझ रहे कि नहीं? So कोकोनट में दोनों टाइप के एंडोस्पर्म मिलेंगे आपको, ठीक है? और वो ड्राई वाला रहेगा आपका, उसके अंदर आपका पूरा का पूरा न्यूक्लियर इज गेटिंग कन्वर्टेड टू सेल्युलर. Because ultimately it has to convert to cellular. The primary endosperm cell divides repeatedly, forms a triploid endosperm tissue. The cells of this tissue are filled with reserve food material used for nutrition of the developing embryo. यहाँ पे function आ गया endosperm का. In the most common type of endosperm development, which is the most common type? Nuclear. The PEN undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs, cytokinesis, and the endosperm becomes cellular. Right? See again the coconut water example is given. It is nothing but free nuclear endosperm, the water, and the surrounding white kernel is the cellular endosperm. And obviously endosperm use अगर हो गया या नहीं हुआ, that will make the seed as endospermic and non-endospermic. Something again what we have covered in morphology, right? So if the entire endosperm is taken up by cotyledons, so do we find an endosperm in that? No. So that becomes what type of a seed? Non-endospermic seed, right? Example pea. Right now, does the seed of the pea has two parts? Those two are cotyledons. They have taken up the entire endosperm. So it's a non-endospermic seed. Right now, so pea, groundnut, beans, all are non-endospermic seeds. Or if endosperm persists in a mature seed, if endosperm is found, then it is what type of a seed? Endospermic seed, like castor and coconut. Right? So, according to endosperm, present or absent? नहीं. Used up or not used up? Not present or absent. Present तो है ही. Okay. Used up or not used up? It is accordingly. Use up हो गया तो non-endospermic, नहीं use हुआ तो endospermic. ठीक है? So that is about development of endosperm. The second is development of embryo. Now before I start development of embryo, just to remind you something that we did in the first lecture of standard 11th, morphology का chapter का first अपना chapter. The embryo is something like a straight line which is called embryo axis. What is embryo axis also called as tigellum? The embryo axis has three structures: plumule, radicle and what is this called as? First node. This is called as first node. और first node पे attach रहेंगे cotyledons. Cotyledons के ऊपर वाला space होता है epicotyl और उसके नीचे वाला space होता है hypocotyl. तो axis पे कितने structures हैं? 5. ऊपर से start करेंगे तो plumule, उसके बाद epicotyl, उसके बाद first node, hypocotyl और फिर radicle. This is your embryo axis. This is your embryo axis. Okay. Now embryo का development obviously will start from zygote. The zygote will develop as a proembryo. The proembryo will become a globular embryo. The globular will become a heart-shaped embryo and then become a mature embryo. जो diagram हमने छोड़ दिया था ना, refer to that diagram please. Can you see the stages of embryo development? Globular, heart-shaped and mature. देख रहे हैं? Globular, heart-shaped and mature. ये stages of development. ठीक है? It's an NCERT diagram, yes. Now, they have given the diagram, but they have given a very poor explanation. Right now, you need to look at it a little properly. Now, if you look at the zygote here, towards the bottom, please. Can you see zygote undergoing a horizontal division here? Now, this is the proembryo. This is the proembryo. Now, we are seeing two cells here. कुछ लेबल नहीं किया हुआ है, right? But ये जो ऊपर वाला है ना, जो ऊपर वाला cell, it is basal cell, right? और नीचे वाला जो है, it is a terminal cell. Basal cell and terminal cell, right? Now, listen to it. It's not required for us. NCERT में description नहीं है. It is only for our understanding that how this structure is developing. Please. ठीक है? Now ये जो basal cell है, उसका repeated division होगा, right? So I'll just focus on the basal cell. Can you see the structure? This has come from ये वाला cell. And can you see this entire structure has been labeled as suspensor. This entire structure has been labeled as suspensor. So ये basal cell से आपको कौन सा structure मिलेगा? Suspensor cell मिलेगा आपको. ठीक है? Now, if we look at suspensor, suspensor has two ends. Can you see a larger cell at one end and this one at the last end, right? Now, so the one which is the larger one is called haustoria, and the one which is the last one at the other end is called hypophysis. So ये किसका डेवलपमेंट कर रहे थे हम लोग? बेसल का. Now terminal का division. Terminal starts its division. Right now, imagine yourself cutting an apple. The first division, is it vertical? The second division is also vertical but at right angles to the first one, thereby creating 4 slices. So 2 vertical divisions will give rise to a 4-celled structure. उसको quadrant बोलते हैं. उसी 4 slices को साथ में रखो and take a horizontal cut. Will that give rise to 8 now? That is called as octet. So this is how it starts dividing, right? And slowly and gradually it will now become first a globular mass. It will become a globular mass. So that is the globular stage of the embryo formation. It's the globular stage. अच्छा, this is the globular stage. ठीक है? Now globular stage में अगर एक तरफ से छोटा सा indentation आ गया तो क्या हो जाएगा? वो heart shaped हो जाएगा, राइट ना? दिख रहा है यहाँ पे? So ये globular है, यहाँ से एक indentation आ रहा है. अब यहाँ से indentation आ गया तो ये क्या हो गया? heart shaped structure हो गया. अब ये indentation बढ़ते जाएगा. This indentation, this is a heart shaped structure, right? As the indentation keeps on increasing, will it become a U-shaped structure now? That is called a horseshoe-shaped structure. That is called as a horseshoe-shaped structure. This is a mature embryo. So mature embryo का shape क्या है? Horseshoe-shaped structure. Mature embryo का. So from a globular, it goes to a heart shape. It goes to a horseshoe shape. What is the horseshoe shape? It is the mature embryo now. In a mature embryo, please, if this is a, let me make a mature embryo once more. Okay, in the mature embryo, please listen to this very carefully. It will help us in identify a diagram. ये जो दो structures दिख रहे हैं अपने को, these are cotyledons. These are cotyledons. Can you see this point here? This is plumule. ये वाला point दिख रहा है अपने को? ये radical है. Radical के ऊपर वाला पार्ट क्या होता है? Hypocotyl. So this region is the hypocotyl. Right? Now look at this diagram. Here. Now look at this diagram. This is a horseshoe U-shape turned upside down. Can you see these are 2 yuga strands? What are that? Cotyledons. What is this tip? Plumule. सबसे बेस में क्या है? Radical. और radical के ऊपर वाला पार्ट क्या है? Hypocotyl. Are you able to understand this diagram, please? Right? Now this is a dicot embryo का structure. 2 cotyledons ना, obviously it's a dicot का structure. So यही structure को उन्होंने उल्टा कर दिया है. These are two cotyledons. This is the plumule, this is the hypocotyl, this is the radicle. Now, looking at this structure and looking at the axis, tell me what is missing in this diagram. Epicotyl. Epicotyl is missing. What is the position of epicotyl on the axis? Between plumule and first node. Barabar hai? So now, as the embryo starts growing, the plumule will give rise to? Get lost. Bada guess what? Plumule will give rise to root. Huh? Stem, right now? Shoot. Positively photo. Positively rising up, right now? So जैसे प्लूम्यूल बढ़ेगा, क्या प्लूम्यूल और फर्स्ट नोड के बीच का डिस्टेंस बढ़ते जाएगा? तब एपिकॉटल आएगा. इसलिए आपको यहाँ पर एपिकॉटल नहीं दिख रहा है. प्लूम्यूल बढ़ने के बाद एपिकॉटल इसमें आएगा. Right? Have you understood the hypocotyl here, please? That is why I had to remind you of this diagram of what we have done in the first lecture of morpho. Look at the development of embryo. Embryo develops at the micropylar end of the embryo sac, where the zygote is situated. Most of the zygotes divide only after a certain amount of endosperm is formed. Okay, we've already done that preceding. Though the seeds differ greatly, the early stages of embryo development are similar in both monocots and dicots. Right? The zygote gives rise to a proembryo, subsequently the globular, heart-shaped and mature. What is the mature shape? Horseshoe shape. A typical dicot embryo consists of an embryonal axis. What is the embryonal axis called? Tigellum. What are the structures on the axis? Plumule, first node and radicle. And from the first node you have two cotyledons. The cotyledons are coming from the first node. The part of the axis above the level of cotyledons is the epicotyl, which terminates with the plumule or stem tip. The cylindrical part below the level of cotyledon is hypocotyl, that terminates at its lower end in the radicle or the root tip. But in this structure, can you see something below the radicle? Root cap. अगर radical से root आता है, तो root cap कहाँ से आता है? Radical last point है आपका axis पे. Is the radical last point of the axis? Radical is giving us to root. It is coming from the hypophysis. Root cap comes from the hypophysis, suspensor wala cell. Is the hypophysis cell the nearest to the embryo? So, our conclusion for root cap, is root cap an embryonic or an extra embryonic structure? It's an extra, it's an outside embryo structure. That's why we will say root cap covers the radicle. It's a structure from the outside. The root tip is covered with the root cap. Now this is a monocot root, right now? Embryos of monocots possess only one cotyledon, and that is known as scutellum. We already done that. In the grass family, Poaceae, the cotyledon is called scutellum, that is situated towards one side of the axis. ये आपका axis है, ये radical है, sorry plumule है, first node है, radical है. So your large cotyledon is situated on one side of the axis. It is situated on one side of the axis. This large cotyledon is called as the scutellum. And the two ends, radicle and plumule, we have studied. Plumule is having a covering called as the coleotile. Radicle is covered by coleorhiza. Right? एकदम nostalgia, morphology वापस आ गया एकदम to haunt us back again. Done? Right? So we have done through now development of embryo and endosperm. So post fertilization we are left with now development of ovule to seed and ovary to fruit, and that will help us to finish the chapter next time. Right? So next time we will finish the chapter and then move on to another bigger headache, ecology. Yes. हाँ, 3 चैप्टर इकोलॉजी में. क्या 12? Obviously 11 तो हो गया खत्म. I am not looking at that book again now. नहीं, I have to look. Frog बाकी है ना, cockroach and frog is still remaining. बाकी है. I beg your pardon? Hollow folio, greenish structure. हाँ? कौन सा टिश्यू? टिश्यू पार्ट है. राइट? ये बार का जो 12th स्टैंडर्ड का बैच है, मैंने उनको बहुत सिंपल स्ट्रेट एडवाइस दिया है कि जो चैप्टर्स डिलीट हो चुके हैं ना, 6 चैप्टर्स, उनको आप छोड़ दो, लेकिन चैप्टर में से जो डिलीटेड पार्ट है उसको मत छोड़ना. कुछ चैप्टर्स में से कुछ पार्ट, राइट ना, जैसे कि टिश्यूज में से पहला पार्ट और सेकेंडरी ग्रोथ डिलीट हो चुका है, तो मैंने उनको बोला है कि जो चैप्टर में से डिलीटेड पार्ट है वो मत छोड़ो, राइट, क्योंकि उस पर अभी भी ग्रे एरिया है. सिर्फ डिलीट 6 चैप्टर्स छोड़ दो, बाकी कुछ मत छोड़ दो. उनको तो ऐसे ही बोला ये बार मैंने. ठीक है, डन. Those are free from the hypophysis.